Milenkovic.
Thanks, Prasad. So I moved the rank aggregation part a little bit towards the end of the title, indicating that I will mostly focus on correlation clustering. And since this is a big data workshop, I will also try to bring in some data and hence the subdued emphasis on rank aggregation. And this is joint work with a few of my collaborators, postdoc Greg Pulio, Amin Ahmad, and our resident bio expert, Jianma. So what is the interest or the idea behind this talk? Very recently, you have observed that there has been quite a lot of interest in community detection. And different people or different research groups called different problems community detection problems. But you can think of community detection problems as basically arising in the study of social structures or social networks, where basically the idea is to identify tight-knit communities by defining a community in terms of the density of connectivity of entities in the group or people in the group. And here is an example, a very clear example of a karate club graph where you have nodes that indicate people, members of the club, and then two different types of communities. And it's clear from the picture that red and blue form different communities because of the density of the connectivity between entities. Other areas where community detection has made appearance is in bioinformatics. We all have some vague understanding that there are communities of interacting entities that are called genes that are jointly responsible for governing or controlling some function in the cell. And in the last 10 years or so, there was a lot of interest in terms of trying to find communities that are related in terms of either similarity of function or related in terms of similarity of sequence or some other property. But another community that you may have seen quite frequently is collaborative communities in mathematics. And this is a very nice drawing that Ron Graham made as a tribute to Erdős, who is a very centrally focused entity in this community. So what is our interest? And the thing that motivated our work is a new paradigm which we call community detection, functional community detection, is particularly in cancer genomics. And it's a very interesting new paradigm because it defines community in a very non-standard way. And let me really quickly explain to you what is a community, or what we would call from now on a pathway in a cancer cell. So what happens in the progression and onset of cancer is basically that certain genes get mutated or replicated. So you have different copies, copy number of the genes. So for simplicity, let's just assume the genes get mutated. There is an uncanny ability that cancer has that it targets only genes in certain pathways or modules in order to preserve energy. And basically the idea is if it can mutate one gene, which is crucial for one module, it doesn't have to touch the other genes. And then what happens is you would see modules where only one gene is mutated, and hence you basically strike out one key player in a community, you attack a member of the community, you take out the whole community, so you don't have to really attack multiple members of the community to disrupt the pathway. So this is what recently came to be known in the bio community as the so-called mutual exclusivity principle. And it says that when you look at the real data, at most one mutation occurs per driver pathway in cancer. Another thing that was observed is that cancer tends to attack a certain number of pathways, and it's important that all these pathways are covered, which means in the majority of patients you will see the same modules attacked, maybe in potentially different ways by attacking different genes, but you will have this coverage property as well. And the third thing that biologists notice is that the modules that are attacked are usually fairly small, and this is what we call bounded pathway size problem, or a pathway limitation to include, let's say, anywhere between 6 and 15 genes. That's true for most pathways, but not for all. So the question is, when we were approached by some people that work in these, on these problems, how do we actually identify these functional pathways by using methods from community detection? And the first thing that came to mind when we saw the problem is to invoke ideas that have been around in the computer science community for almost 15 years. And it's a work that was known under the name of correlation clustering, which may be a little bit misleading, but in general it refers to a set of techniques that are concerned with optimization on graphs. So correlation clustering was introduced by Bansal, Bloom, and Chawla. And in its most elementary plain vanilla formulation, it says that you're given n objects. You can view them as nodes in a graph. And for some of these objects, you give a judgment or an assessment of how similar or dissimilar the objects are. So for example, here you have a graph, and objects that are similar are connected by blue edges, those that are dissimilar by orange edges. So we can assume that the color of the edges indicates a plus or minus, or infinity or a similarity or dissimilarity. And the question now, the problem of interest, is partition the vertices into clusters or community so that you minimize the number of positive or blue edges going between communities or clusters, and at the same time minimize the number of negative edges or disagreeing entities or dissimilar entities within clusters. So this is an obvious solution for this problem. You will just put 2 by 2 and then 1 vertex here, because you only have blue edges within clusters, orange edges between clusters, and unfortunately these 3 edges also go between clusters. So this is what is plain vanilla correlation clustering. And correlation clustering is an optimization on graph kind of problem, but it's also some form of agnostic learning, because we were never told in advance what the number of communities is, which is very important for bio-related problems. And it's related to a host of very interesting questions which deal with optimization on graphs, such as balanced graph partitioning, edge partitioning, rank aggregation, which was supposed to be the second topic in this talk. I'll comment on it a little bit later towards the end of the presentation. So what is known about this correlation clustering, and what are the basic results we had so far? Finding the best clustering is obviously NP hard, so we're interested in approximation algorithm. When the graph has a general structure, we have best known approximation ratio of the order of log n. For complete graphs, Eilon, Chariker, and Newman proposed as early as in 2005 a randomized expected 2.5 approximation algorithm. And the work that we are going to be mostly interested in is the work by Charikar, Guruswamy, and Wirth, who gave a deterministic 4 approximation algorithm. There have been some results, very recent results from this year, that improved this constant to 2.6, but that's not going to be that relevant for this presentation. So what is even more important and more interesting for our setting is the fact that you don't have to stick to the plain vanilla description of the problem. You can also assume that the edges, rather than having a plus-minus label, also have weights. And the weights are assumed to be negative. And you're paying a cost, let's say, denoted by omega E plus, when you place this edge between clusters. So each edge misplacement has its cost, and the cost of misplacing an edge by placing it within a cluster is denoted by omega E minus. So by choosing those weights, you can control how much an edge is important in your graph and how much do you want to emphasize where this edge should belong in the cluster structure. And you can also control, in some sense, the topology of the network, because basically you can set both weights to be equal to 0 for some edges, which would then imply that that edge is completely gone from the graph. And most of the results so far have focused on weights that are highly structured insofar that they satisfy what is known as the probability constraint. So for each edge, we assume that the cost of the negative or positive misplacement sums up to 1. And for this, we know that there is a randomized 2.5 approximation algorithm due to Eilon, Chariker, and Newman. And there are some results that tell you how to go from unweighted problems and get algorithms for weighted problems. And the question is, given this very quick overview of the history of the problem, do any of these methods work for the community detection question we had in mind in cancer genomics? And the answer is, based on the observations that we had before, what do we really need for community detection in cancer genomics? So here is an example, and this is real data from the TCGA database, or the Cancer Genome Atlas. And it lists for something that we call a pseudo pathway. It's just an example where you have genes indexing the vertices, and you have edges between genes that tell basically in how many patients where both genes simultaneously mutated. So for example, this number here would say that only 2 out of 26 patients had a mutation in this gene and this gene in the case of breast cancer. So mutual exclusivity really can be handled in the correlation clustering setting if you allow very high negative weights for edges that would correspond or capture this mutual exclusivity that we know exists in cancer genomes or cancer genes. The idea of coverage, or how to accommodate coverage in this question, is basically through the weights of the positive edges. So you would have to assign in a very careful manner the weights, positive weights of the edges in such a way that you get this nice coverage that most patients will have mutations within one group of genes, so that you have this x-axis which correspond to the number of patients, showing this nice coverage of all 60 patients with respect to these 6 mutated genes. And then in both cases, we would need some kind of way to handle bounded cluster sizes, but really what we need is soft bounding, because some clusters can be large as long as there are not too many mutations in those clusters. So how do we do this? So we really couldn't apply any of the existing algorithms because, number one, none of the correlation clustering methods are able to deal with bounded or soft bounded cluster sizes. Second, the weight values that we need were not covered by any of the solutions that exist. So we really had to redesign and develop new algorithmic solutions that I will discuss in the next few minutes. And then I will show you some results how we apply these algorithms in the analysis of breast cancer patient data. So to explain the algorithm, let me just quickly explain to you the algorithm from which we started, which is designed for the unweighted case by Charikar, Guruswamy, and Wirth. And it's a very simple 2-step algorithm. The gist of the most important aspect of this algorithm is basically to show that it works. But the algorithm itself is pretty straightforward. So we will introduce one variable for each edge. And here we are denoting the edge in terms of its 2 endpoints, and one should serve or be interpreted as saying that those 2 vertices or the edge between them belong to different clusters, and 0 would mean that the endpoint edge vertices belong to the same cluster. So you would start with an integer program where you're trying to minimize the total number of misplaced edges, basically positive edges between clusters and negative edges within clusters. And this inequality here will basically just tell you that if you put 2 edges, adjacent edges in the graph, and there is obviously a 3rd edge between them, you would have to complete the triangle. You cannot separate entities, these vertices in the community. So if you relax this to an LP, you get solutions which tell you something like fuzzy clustering. You will only know if U and V are likely to be in the same cluster or not likely to be in the same cluster. So what you need to do is perform a second step, which is the rounding up procedure, where you take the results of the LP, and recall we said that XUW0 means the U and W. Yes, please.
Sorry, before you go further, can you go back to the algorithm?
Sure.
We can understand So the edges the labels are assigned to you, right?
Yes, we determine the. This is right now. This is Charikar, Guruswamy, Wirth, which doesn't have weights.
But I'm saying what is given to you are the labels on the edges.
Yes.
You want to find the labels of the nodes, right?
Sorry.
And you want to.
Yes, exactly. You want to find the communities, right?
So why do you optimize over the XUV? Isn't XUV the edge label? No. XUV is not
this here tells you which edges are positively labeled. This here tells you which edges.
XUV is not the label on the edge.
No, XUV tells you if they belong to the same cluster or not. So if XUV is 1, you should interpret it as U and V belong to the same cluster. That edge is placed in one cluster. It doesn't go across clusters. Any other questions? Okay, so once you have this, so basically what you get is soft assessments. Are 2 vertices supposed to be in the same cluster or not? And then you need to do some rounding up because you're going to get values that are not necessarily 0s and 1s. So this is what the rounding up procedure does, and it tells you if a value of what you should do is randomly pick an element, pick an arbitrary element, and then check what the value of its neighbors is. So if the values of the linear programming outputs are very small, maybe you should put all those neighbors of the selected pivoting vertex in the same cluster or in the same community. And when you do that, you should check that the average weight of that community or that cluster is not too large. If it's too large, you just output a singleton. If it's not too large, if it's less than or equal to t/4, then you form a community by merging all the neighbors that you found sufficiently close to the vertex you picked. So, yes, please.
Isn't 4 times a very bad bound?
It's a very hard bound to get. It's actually, the result is very good, but from practical point of view, it's obviously not guaranteeing much. But I will come back to that later if I have the time. So this is the result by Charikar, Guruswamy, and Wirth. And I mentioned that there were some improvements recently, but it's still 2.6. So how do we handle the bounded cluster size and the new weights? So the bounded cluster size we are going to handle by first introducing a size parameter for the clusters, capital K, and then we are going to assign a special parameter for each vertex V, which is going to be non-negative, mu V. And if this vertex here ends up in a cluster of size that exceeds K by this much, where YV is the excess cluster size, then we are going to penalize this vertex V in terms of a cost that reads as mu V times YV. If we set all these extra penalties to 0, we don't really deal with cluster size constraints. And if we set them to be large enough, larger than the maximum positive weight, we will always get very hard constraints. Basically, we will always encourage having clusters of size less than or equal to K. And this will allow us to handle soft constraints, basically allowing some clusters to be large if they have very few negative edges. And how do we deal with the weights, and how do we incorporate the cluster size constraint? So the weights are very easy to put in. All you need to do is multiply each of the indicator variables with the appropriate weights. Now we have this extra term in our LP, which tells you how much do you penalize oversized clusters. This extra constraint tells you basically how the clusters have to be kept bounded or softly bounded. And the interesting thing is now that you have a rounding up procedure where you can no longer just rely on saying that vertices should be lumped together if they're, let's say, estimated distance from the LP is less than 1/2. Now you have a threshold alpha, which will tell you when you should group elements together with the pivot vertex, and that threshold was 1/2 in the original unweighted algorithm. In this algorithm, it will have to depend on the choice of the weights and on the way we try to impose those cluster size constraints. So here are the results we have. So we have constant factor approximation algorithms that work in the following cases for these weights. So these are extensions of the weight results that existed in the literature. So we basically now allow, to just skip through this because I'm running out of time, we are allowing very large values for the negative weights, which means we can discourage two vertices being put in the same cluster quite heavily. And this is what we really need to enforce mutual exclusivity in a very strong way. And if you let the weights grow, you basically go back to a problem which is known as the cluster deletion problem by Shamir, Sharan, and Sur, which is interesting in its own right. And this is very interesting because we couldn't get the similar approximation bounds for when we allow very large positive weights. So what we basically have to deal with is large negative weights, and this is what we obtained as the results. And how to choose the optimal alpha for the rounding up procedure is really a big mess. I'll skip this, but it boils down to finding the solution for a cubic equation. And for some cases, the solution is fairly straightforward. For most of them, it's rather complicated, but it tells you that we can do something like a uniform approximation for the approximation ratio, which depends on this parameter tau, which is tied up with the choice of alpha and the weights through this messy equation here. So let me then go ahead, and these are the approximation ratios. They're not they don't appear to be that good. It's 6 or 6.2. But here are other ways to do this. You don't have to put the size-bound constraints in the LP procedure. You can actually do this control of the size of the communities in the rounding up procedure. So you just solve the LP, and if the community you get by rounding up is too large, just break it into sub-communities, and there are different ways to do this. And these algorithms can also be provably shown to give you constant approximations, and they actually work better in practice than the previous algorithms. And since I'm running out of time, there are other ways to do this. You can do randomized algorithms similar to this very well-known CC pivot algorithm, and it relies on using some interesting results from edge sparsification, as I call it, by Hel, Kirkpatrick, and Gabo, which I can discuss with you online. And what I want to show you now is, since we have only a few minutes, some of the really interesting results that you can get by applying the last algorithm I talked about to benchmark data that is available at the Cancer Genome Atlas. So far, there were two algorithms that tried to exploit this mutual exclusivity property for community detection. They used some integer programming methods to try to come up with the communities. One method is particularly well known. It's called the Dendrix method. And the Dendrix method tried to impose only the mutual exclusivity principle by performing certain steps in the optimization algorithm, which I won't discuss right now. But what we did is we tried doing the analysis using correlation clustering, and we worked with data of 504 patients, and this is just for breast cancer, which had a total of roughly 9,000 mutated genes. And what we came up with is some quite interesting results. So let me show you just two of them. There are many, many of these pathways that can be identified in this way. This is a set of genes in a community or in a pathway which basically tell you very nicely the different forms of breast cancer that are out there. So it's pretty well known that patients that have a mutation in this gene, which is quite dominant, the largest number of breast cancer have mutations in this kinase gene, those patients would respond really well to hormonal therapy. The ones that have mutation in CDH1, you can see that there are very few patients, based on the clustering we did, very few patients that have a mutation in both. And that very much confirms the different type of cancer results that are out there. These patients are so-called triple-negative breast cancer patients. They have a very poor prognosis because they don't respond to treatment, especially hormonal treatment. So one of the goals, and I'll stop in a minute, is to basically take these algorithms and apply them to patients before and after treatment. Because what turns out to be the case is that most cancer patients, once they're treated with one drug, will develop mutations in a different pathway. And it's really a disturbing thing because you're treating a patient for mutations and trying to prevent wrong expression in one pathway, but another pathway gets compromised. So by doing before and after community detection analysis, one would hope to see basically how the pathways shift. And I'll skip the rank aggregation problem completely. It's very much related to what I talked about. So if you're interested in talking about this online, I'll be very happy to do so. And I would like to thank you all for being here.
We have time for one quick question by Jacob Sexton.
So one issue with ELP is that you have n squared bar with an n cube constraint, right? So like if you want to scale for more than 1,000 n, that would probably be really slow. So I was wondering, how good does just the rounding step perform without ELP? So sort of the greedy part of the...
So we tried the CC pivoting thing.
Yeah.
It's not working well. And the one thing that I didn't mention is you always do filtration. You take genes that are mutated in very few patients out. So you never work with the 8,000. You boil it down usually to 600, 700. But it's still a large LP. Is it important to get it done in real time? Right now, probably not, because it's mostly exploratory.
Yeah, so I was just curious, if you get, so if the maximizers, if the maximum of the relaxed version is within, let's say 4 times the unrelaxed version, can you say something about the maximizer? Do they overlap with,
That's always the problem with this. You can only say when you apply it directly. You run it and you check what how the results look like, but there's no guarantee that
the
there's
partition
any
is what you're looking for, yes?
Yeah.
Not
And
the, not the
there may be a lot of optimal solutions, and they may look very different. But that's the problem you have with all these problems where you use approximation algorithm, and that's all of bio. Sorry.